Hey guys, welcome to our last topic in chapter 14. In this topic, we're going to address the origins of chloroplasts and mitochondria a little bit and talk about the evidence that we have that supports our theories of the endosymbiont theory and how these organelles have led to the development of life on this earth and life as we know it. So here's our topic outline. It's pretty simple. We're just going to talk about the origins of chloroplasts and mitochondria. And you only have one objective for this topic. It's a short topic, but it's really important. So I expect you to understand these and be able to discuss this concept, but I don't expect it to be the bulk of the understanding coming out of this chapter. So it's important to understand, but the main focus for you should be in the first two topics. All right, so let's go ahead and get started. We've already talked about the endosymbiont theory, which is that we have this primordial bacterium, pre-eukaryotic cell, whatever you want to call it, that was really large, and it engulfed the mitochondria, it engulfed the photosynthetic bacterium. And rather than digesting these bacterium, they actually developed a symbiotic relationship where they helped each other and became the cells that make up you and I and make up everything else that's a eukaryotic organism on this planet. So let's talk a little bit about these organelles. We keep talking about the fact that there's this genome, they have genes, they have all this stuff. And most of those original genes that any bacterium would have to survive on their own have actually migrated to the nucleus of the eukaryotic cell. So the eukaryotic cell, or the nucleus of the eukaryotic cell, has most of the genes for these organelles. However, mitochondria and chloroplasts still have their own genes. Mitochondria have a small genome, about 37 genes, and chloroplasts have about 120 genes. And the genome of the chloroplast is actually really similar still to the cyanobacteria, which is is a photosynthetic bacteria. So it's a pretty good relative of what was the ancestral chloroplast for us. And these gene products that are needed for these organelles that are actually in the nucleus are exported from the nucleus to the mitochondria. So when we talk about like mitochondrial ribosomes and everything like that, those are actually made in the nucleus and transported through a variety of vesicles to the mitochondria. And we'll talk about that a little bit later in this semester when we talk about intracellular transport. So at the moment, it's just really important to understand that they do have their own genomes, but they're really small, and that a majority of the products needed for these organelles are made in the nucleus and then exported. All right, so let's talk a little bit about ATP synthase. Obviously, both these organelles use this machinery. But we want to know, how did this machinery possibly evolve at first? Well, the first thing you have to think about is back to the environment that these cells first existed in. The early environment would have been really acidic. And so any cell that had the ability to regulate the pH of regulate its own pH and maintain that environmental balance would have been very evolutionary useful, because they could have lived in a very acidic environment but still maintained the internal pH that's needed for proteins to function. Because don't forget, proteins and DNA even are very sensitive to pH, and so it's really important that the cells maintain that, otherwise they're going to not be able to function. And so the cells, we believe that the first primitive bacterium evolved with an ATP-driven proton pump. So using ATP, they would pump out protons. Now this seems weird, because why would we use ATP to pump out that proton? But remember how important pH balance is to the cell. Then we believe that the next step along was these electron transport protein pumps that would move these protons out. And eventually the whole idea of the electron transport chain, and then using that ATP synthase, because remember, ATP synthase can work in both directions, developed together to form the oxidative phosphorylation chemiosmosis electron transport chain that we've talked about. And so we believe that this is how it's processed through these three different stages. And so that's why part of our evidence for this process is that we believe that because the pump can work in both directions, and we see this in modern day bacterium still, that the ATP synthase works in both directions, as we talked about in the other topics. So now let's talk about the development of photochemical reaction centers. More than being able to balance their pH, photochemical reaction centers was a huge advancement, evolutionarily speaking. So what this did is it allowed organisms for the first time to not rely entirely on their environment for life. They were able to harness light energy through photosynthesis to be able to do this. Now, the first photosynthetic reactions used H2S instead of H2O, and that's because H2S has a higher redox potential. What this allowed for was for only one photosystem instead of the two photosystems we talked about in topic 2 to be utilized, because all we needed to do then was generate the NADPH. And so it's really important that we recognize just how important the development of these photosystems was, because we allowed everything to be on their own and allowed for this development. But even more than that, when these plants and photosynthetic bacteria evolved to use H2O in photosynthesis, this is when oxygen began being part of our environment that resulted in how we, in the environment that we know. And so it's important that you understand how this works and how important the development of these photosystems are. So just as important as the ATP synthase was to pump out protons to balance out the pH, it's important that we understand that being able to not depend entirely on the environment of that an organism is in was a really big evolutionary advantage. And so we can see this where I talked about the how the photosystems led to the accumulation of oxygen. So once water splitting became part of photosystem 2, then the accumulation of oxygen began. And as you can see here, this map tracks a little bit about how life has evolved through the accumulation of oxygen. And remember, without oxygen, we, as we know it, would not exist. So it's really important that we understand how chloroplasts and mitochondria have driven the world that we know today. Now we have an example of this, and we see a couple examples through extremophiles, and we can use these to kind of gauge as to what old bacteria and the first life on this planet would look like, because they live in very extreme environments. And these environments are very similar to what we believe Earth looked like way back before the environment that we live in today has been established. And one example is Methanococcus. It lives in very high temperatures. It relies on nitrogen gas. It grows in inorganic nutrients. It doesn't need light. It doesn't need oxygen. A lot of what we think life on Earth looked like back then. And in order to survive, they use an electron transport chain where they transfer electrons from hydrogen to CO2 and release methane gas. And this is all occurring through proton pumping. And so this kind of gives us a sense of what these primitive electron transport chains and chemiosmosis pumps and the ATP synthase would have looked like in an environment that didn't have oxygen and didn't have all the readily available nutrients we've talked about. And so it's really important that you understand that Methanococcus is one of those bridges to the past that we use to help us understand the evolution of these processes. So that's the end of this short topic. Please let me know if you have any questions about it, but it's really important that you understand our evidence that supports the evolutionary theories behind the endosymbiont and chloroplasts and mitochondria and how they've evolved. And if you have any questions, please let me know.